before. There's going to be an RNA-Seq folder available to you. We have a number of different outputs that you can take a look at. The two that we're going to be focusing in on are these two example folders here, this HT-Seq, DE-Seq, and antibiotics and Acinetobacter example. The example that you can submit if you would like to follow along and do your own submission is found in the HT-Seq, DE-Seq example folder under Orientia carp. So going into that folder, you'll find this text file, which opening this up will give you some SRR IDs and then what actually was done to those samples. So the conditions. First, so I'm going to leave this over in this tab. You can open it up. So let's open up a second tab, go to Tools and Services, and then under Transcriptomics, we have RNA-Seq Analysis. So the RNA-Seq Analysis page is going to be one of the more complicated input pages. We have some of the same sections as the other services, your selected libraries, paired reads, single-end reads, SRI accessions. The two additional sections that are not found in other services are your groups and conditions box down here. And this is also connected to the contrasts box in the bottom. Because one of the major things that you can do with RNA-Seq is differential expression. You want to figure out which regions, which transcripts are upregulated or downregulated when comparing two conditions. Filling out this form, the first thing you want to do is fill out the parameters box. Currently, we have 3 strategies available. As Rebecca had said, we're going to be improving this over time, so we might be adding some more strategies in the future. The differences between these, Tuxedo pipeline is a strategy that utilizes cufflinks. Tuxedo is a published RNA-Seq pipeline that has a lot of support for it, but it's also one of the older tools. So a lot of new stuff has come out that will give some different, more interesting results, but cufflinks is still useful in some circumstances. HT-Seq, DE-Seq2, it uses 2 of the newer tools that seem to be more state-of-the-art in the RNA-Seq world. HT-Seq is one of the abundance estimators. I'll go over more in detail on some of the parts of the pipeline. And then DE-Seq2 is the differential expression program. Host HiSat2 is the strategy that will be used whenever you're looking at host RNA-Seq, regardless of if it's bacterial or viral infected. This is the standard one. So the first thing you're going to want to do, though, even before selecting a strategy, is selecting your target genome, because depending on your target genome, it might auto-select the strategy for you. So safest thing to do is just fill this out. For this example, we can, actually don't remember the genome ID off the top of my head, but since we already have the output available to us, if you click on that job result, you can see, first of all, this folder that has a number on it, that's going to be the reference genome ID that we're going to be using. It's also found in the job metadata that's been pointed out before. So over here, this breadcrumb at the top, we have the BV-BRC workshop folder. Go into RNA-Seq, HT-Seq, DE-Seq example, and then Orientia carp HT-Seq will bring you to the file that has the SRR IDs. And so we're going to be entering in each of the SRR IDs in this file.
Just really quickly, does anybody know what Orientia is? Orientia tsutsugamuchi. It is the causative agent of scrub typhus. So we've gone beyond endosymbionts, and now we're talking about boring actual pathogens. It's transmitted by ticks, and in parts of Asia, it's a real problem. Actually, it's mites. Arachnids, it's vectored by arachnids. I just like to give the backstory, Clark.
No, that's fine. Please fill it in. So after selecting the target genome, you can select something related to Orientia tsutsugamuchi, preferably the carp strain. That will work. And we can select the output folder to give the output name. The groups and conditions. In order to do differential expression, you have to assign each of your samples a condition of some sort. So we can just turn this on. You can just do RNA-Seq and just get your abundance estimates of transcripts or something. You don't have to do differential expression, but differential expression is one of the more powerful aspects of it. So that's going to be part of this example. So turn on the groups and conditions. If we go back to that file, you can see that there are 2 descriptions. We have infected with Orientia carp, and then infected with Orientia UT176. You can arbitrarily name your conditions whatever you want. You can just say condition 1, condition 2. For this example, I'm going to be using the strain name, CARP N UT176. So you can just go over here, type in the name of the condition, and then pressing the plus button adds it to the groups and conditions library right here. Just going to do that with the other one, UT176 plus, and now there's another condition. And I don't believe that there's a limit to the number of conditions or files that you can set, but just keep in mind, RNA-Seq is one of the services that can take the longest to run depending on how big your files are. So you might want to keep it smaller when doing these analyses. So now that we have our conditions, we can go ahead and select each of the SRR IDs. And before we press the arrow to add it to the selected libraries, we need to add the condition. You're going to go through the same process if you have or want to use FASTQ files. You would just select the FASTQ file, then add the condition, and then click the arrow over. So this one was infected with Orientia carp. So the dropdown, I select carp, and then press over. And as you can see, the SRI ID or the name of the sample, along with a identifier for that condition. So I'm just going to go ahead and do that for each of the IDs in here.
And while he's doing that, the two strains that they're looking at there, one is highly virulent and kills mice very, very quickly, and the other kills them less quickly. So that's why the original experimenters were looking at this. And this came from a paper in PLOS, neglected pathogens.
Okay, so I'll pause for a moment, let everyone catch up. All of these samples have their conditions assigned, and they're in the selected library section. Before I continue, are there any questions? So the question was, how do you choose which one? We're going to submit this job, and then I will go through and actually explain in greater detail what each of these do. Thank you for that question, because I forgot to say, select the HT-Seq DE-Seq strategy, because that will give you the results similar to the ones that we'll be looking at. So if everyone has those up, in order to finish off this page, we're going to be going into the contrasts box at the very bottom. And this is very important because this is where you define what your in differential expression you're going to get the log fold change values. And here is where you define which sample is going to be considered the upregulated versus downregulated sample. So in this case, we're going to select CARP as condition 1, UT176 as condition 2, select the plus button. So in this case, CARP will be considered upregulated. So if you see positive log fold change values, that means that that gene, that transcript is upregulated in the CARP condition. Negative log fold change values indicate that it's downregulated in CARP or upregulated in UT176. This is very important because this part is crucial in your understanding of the actual results. That is it for filling out the form. Whenever you're ready, you can go ahead and press the submit button. So now that this job has been submitted, I'll go into explanation of each of these strategies. So as you can see, we have 3 currently available. I mentioned in the beginning, there's the Tuxedo strategy, which the core software there is called cufflinks. In differential expression, it also uses something called cuffdiff. The second one is HT-Seq DE-Seq. As I said before, the major differences there are it's going to use HT-Seq as the program that does abundance estimation in order to give you your raw gene counts, and then uses DE-Seq as the differential expression program. The similarities between these two are that they use Bowtie as the aligner. FastQC is then output into your workspace so that you can see whether or not your reads were of good quality. And one of the more important differences between these two, the HT-Seq DE-Seq pipeline requires you to have a minimum of 2 replicates in order to actually use it. The program DE-Seq2, you cannot run it if you do not have 2 replicates of each condition. And that's one of its pros, but also one of its cons. You get better statistical power and evidence for your upregulated and downregulated genes. Tuxedo can be run with a single sample per condition. So it allows you to do some quick and dirty estimates, but you also don't have as much statistical support for your genes. And the final strategy, the one used purely for host samples, the major differences for the host HiSat2 strategy is that instead of HT-Seq for abundance estimation, we use StringTie. StringTie also incorporates knowledge of transcripts, which HT-Seq doesn't necessarily do. It also uses HiSat2, which is a splice-aware aligner, And it's pretty much just a wrapper for Bowtie2. Uses that in the background, but incorporates all the different splicing. If you're looking for parity of results between raw copper runs, you would use Tuxedo in order to compare it. But that's also one of the difficulties with RNA-Seq is these updated programs can give you disturbingly different results sometimes. And so it's very difficult to compare them. Let's see. So those are the major differences between these strategies. Just to summarize, Tuxedo, good if you only have a single replicate per condition. And for bacterial RNA-Seq, HT-Seq, DE-Seq2, this is recommended if you have multiple replicates per condition. Also, just for bacterial runs. And then host HiSat2 is going to be the default whenever you are doing host RNA-Seq.
Does HiSat2 used for host gene expression analysis work for bacterial and viral gene expression analysis of host?
To answer viral RNA-Seq, to my knowledge, the only time you're going to be doing viral RNA-Seq is host response. So in that situation, yes, because HiSat2 is made specifically for host alignment. You can use HiSat2 for bacterial RNA-Seq. It's just unnecessary because you don't have to be aware of splice junctions and introns and extrons, et cetera.
And a second question, does HiSat2 require a number of replicates?
HiSat2 does not. I don't remember if we changed the differential expression backend for HiSat2, so you might actually need 2 runs, but I'm going to look into that at some point. So let's go back over to the Orientia CARP folder. Here we have a job result already prepared. So we can click on the checkered flag to descend into those results. And as was mentioned, we're going to be continuing RNA-Seq. One of the better issues with this pipeline is there's a lot of output. And there's a lot of stuff to sift through. So we're currently working on the best way to organize this and display it for you so you don't get lost in these folders. So the first thing we're going to look at, as I said before, is the genome ID folder. Descending into that, we have a number of different outputs. I can step through these very quickly. Just going to click on them and show each file briefly. This gene counts CSV folder is just going to be the raw gene counts. So for each of your samples, these are the raw counts. Go back up by clicking on the genome folder. This file, very similar to the gene counts folder, except this has the gene counts converted into TPMs, transcripts per million. This is a normalization metric in order for you to compare between conditions. So this will be something easier to see once we go and show the heat map, but this is good for comparing sets of your different samples so you can see which genes are upregulated or downregulated. So the question was, can you convert the TPMs into log TPM values in order to bring down the scale of some of those TPM values? That's not currently implemented as something you can quick and dirty do on the website, but it's really easy to just download that file and then do that yourself. Going back up one folder. So here we have this file called heat map genes. This is a file that contains your most highly differentially expressed genes if you have turned on differential expression. We limit it to 50 because sometimes there's just too much information, but these should be the top 50 genes that are most different between your conditions. I'm not going to click on it because these genes are also the ones that are used for the file name in the heat map that I'll show in just a minute. This genome ID underscore report HTML file is a very useful file. It uses this program called MultiQC, which is really good for just combining a lot of different output results. It goes into each of the individual samples and will give you a variety of statistics and incorporate the outputs of a number of different programs. The general statistics table here is very good for giving you an overview of how your samples might be different in terms of the number of sequences that are actually used in the sample, the percentage that get assigned. So if there is a sample that is drastically different from the others, you'll be able to see that very quickly here at this table in the top. So it incorporates, as you can see, some of the FastQC output. And already one of the things we can notice is that these 3 samples have a significantly different number of reads compared to these 3 samples. And if you notice, these 3 samples are the ones that were infected with Orientia CARP. These 3 samples were the ones infected with UT176. So just something that you can notice right off the bat when looking at this report. Continuing on, and just a lot of different stuff that we've seen from FastQC. And here we go. So it also takes the HT-Seq output and then puts it into a summary graph for you. And so again, we already know that we have more reads in the Orientia CARP samples. Here we have the different assignments for what these colors mean. So this is just one of the observations you can make looking at this graph. Our reference genome was an Orientia carp reference genome. And as you can see, we do not have any of the not aligned category for these three samples. These samples were infected with the UT176. And as you can see, there are a number of reads that did not align to the carp reference genome. So those reads are going to be reads that are specific, would only align specifically to UT176. That might be something interesting to go for a further analysis, but just something you can see right off the bat. So there's a report folder. You will also have a folder for each of your conditions. And just to really quickly dive in, here are each of the replicates. And there's just, as I said before, an abundance of information that is sometimes difficult to sift through. So working on making this easier, but if you wanted to use your BAM file for maybe looking in the genome browser, it's possible to do that. We have a number of different other statistics. I'm not going to dive into this. There's just a little too much to go through. This folder, TPM calculator output, is the program that's used to convert the raw gene counts into TPM values. It uses a NCBI program called TPM calculator, just very convenient. This file here is going to be the raw output from DE-Seq2 once it opens eventually. So as I said, this is the raw output from DE-Seq2. We have the name of each of the genes. You have this base mean value. The log2, which less important, but some people might find it useful. The more important column, log2 fold change. This is going to tell you which genes were upregulated or downregulated between your conditions. As I said in the beginning, how you set up your contrast is very important when looking at this file. Negative values, like this line right here, it's -6 log2 fold change. This means it's much more upregulated in the UT176 sample, but this gene here has a positive log2 fold change value. It's much more upregulated in the carp samples. And this is just a general viewer for TSV files, so you can do stuff like sorting, find the most differentially expressed genes just by sorting on it. Then you have your p-values for figuring out whether or not this log2 fold change is statistically significant. And these two files are less important, but this is used in the differential expression heat map, which we'll go into in just a minute. And this library geometry file has some additional information on straightedness of your reads, not as important in a lot of circumstances. So that was all in the genome folder. Metadata.txt is just going to be your assignments for your samples. I'll just dive into it really quickly. So you can see the sample name and the condition that you'd assigned it as a good reference. We're going to skip over this for the moment. We'll come back to this. This is the differential expression output and allows you to see it in a heat map form, which is very convenient. Pipeline.txt, if you, for whatever reason, have any interest in seeing the actual commands that were run in your RNA-seq analysis job, then you can just go there to see all the command line commands in all its glory.
That's my favorite one. I always open that one and look at it in detail. It's like a great read.
Mm-hmm.
Not, but some
great for publications. That's all you want to see is command line.
Yeah, submit that as a supplemental one.
So one of the things that we're trying to embellish a little bit are the different figures that you can generate using RNA-seq output. Again, currently in progress, but some of these are very fun to look at. Given the reference genome ID, we download pathway and system information and then give you violin plots so you can get a sense of the expression distribution of the genes involved in those different pathways. In this particular example, we don't see a lot of a difference in these categories, but if there was a difference, it would be really cool to see one being very different in one condition versus the other. The samples are colored by their conditions. We're working on grouping these together better. So these are very cool to look at. Similarly, this is for different types of system information. This volcano plots file. This is going to be a visualization of your differential expression results. And there are different categories in this plot. The gray ones, which are these guys down here, just means that they are not statistically significantly different between the two conditions. And there is not a large log2 fold change difference in those genes either. Green values have a large difference in log2 fold change, but that log2 fold change difference is not statistically significant. Blue ones are the exact opposite. They're considered statistically significant, but have a very low log2 fold change difference. And the red ones are the ones that you're going to be interested in. Those are the ones that are actually different, and there is evidence provided in your samples to support the idea that they are actually different. So some of these can be fairly interesting. I've seen a number of different graphs where you only get these red dots on one side. So only one of the conditions has upregulated or downregulated genes. This is a very useful image. So the question was, do we provide a workflow on how you can generate these images yourself in order so you can play around with the colors, make it something more appealing to you? And that's something we're trying to put together so that you can take the raw output that we provide you and you can customize it as necessary. Not available yet, in progress. This is the heat map of the top differentially expressed genes from your comparison. These are grouped by their condition. So on this side, we have the UT176 samples. On this side, we have the CARP samples. So as you can see, it identifies the genes that are most different between these two sets. Some things that we're playing with, we had to shorten these identifiers. So in place of this star, you would put the fig bar, genome ID, PEG, etc. These values are TPM values, not the raw gene counts, so that you can actually do between condition comparisons. And something that we're trying to include is additional metadata information on these genes. So for this particular example, if this gene is colored red, that means that this gene is involved in membrane transport. So you can see one of the genes at the very top is also involved in membrane transport. The more metadata there is, the more that will appear in the heat map. For this one, there's just a little bit though. So that's a very broad overview of all of those files. But one of the cooler things, after you do differential expression, you'll get this job result within a job result. Clicking on that brings you to all these JSON files, which we established, very boring, not useful to look at, but you can press the experiment button in the top right corner, and that will bring you over to a different view so you can see your differential expression results in an interactable way. So here's a table that gives you the actual gene identifiers, some metadata for it, like product or a description of the gene. This comparisons column is just the number of comparisons this gene shows up in. So if you have multiple differential expression comparisons, then this number will go up. And up and down just indicates whether or not that gene is upregulated or downregulated. So this is going to be a fairly boring heat map. We can't even cluster this because we only did one differential expression example. So that's when we bounce over. Yes, any question? You can export the heat map. Let's go into that and just confirm that really quickly. Over to the heat map. And yeah, we have this Save SVG button, so you can save it yourself. So let's go back up to RNA-Seq, navigate over to the antibiotics and Acinetobacter example. Click on this job result. Needs to be cleaned up a little bit, but we're just going to dive right into the checkered flag within the checkered flag. Again, click on the experiment button, and that will take us over to the differential expression results. Now, to give background for this comparison, in this example, what are the Acinetobacter examples? Yeah, Rebecca has more of the story for this one.
2 antibiotics and a control, and this was run with Tuxedo as opposed to differential expression. And so the original authors, this was in Acinetobacter baumannii, and they were looking at up and down regulation of genes during exposure to coal, which Jim, I think, would be able to tell us what that antibiotic against marrow is meropen, and colicine, meropenem, and Mueller-Hinton broth.
75% of the lethal concentration. So you want to see what genes get expressed up or down regulated in the organism in response to the antibiotic stress.
The major reason we came over to this example, though, is we have multiple comparisons that were performed. So when we go to the heat map this time, we have multiple rows so that we can actually do something useful like clustering. Click that button, and this is going to group together genes by whether or not they were upregulated or downregulated in those comparisons. One thing that might take you off guard, red in this case means upregulated, green means downregulated. I've had a number of different comments about that, working on changing those colors, or even better, letting you customize which colors mean upregulated and downregulated, or not.
General heat map, for those of you who know RNA-Seq, for some reason, some people said it this way originally, that red's up and green is down.
So the fun fact, this comes from the microarray days when there were cyanine 3 and cyanine 5 color channels for red and green. So that's where it comes from. We have another color scale over here, which was more patriotic, red, white, and blue. That's another popular color scale that people use.
So when interpreting this, cooler things you can do. So each of these columns is going to be a particular genes. Let's see, the bottom one here is going to be the Maripin versus the Mueller-Hinton broth. The one right above it is the coal over the broth. And then this one is a comparison between the two different antibiotics. So some interesting things that you can see. This selection of genes here is going to be downregulated for each antibiotic or upregulated in the broth samples. So if you wanted to see these genes that get turned off when exposed to the antibiotics, you can do as Rebecca showed before, do a selection, and you'll be able to do a variety of different things with that selection of genes for further analysis. I don't think that it will actually show the genes in the column names, or maybe it does. Yeah, there we go.
If you want to annotate your heat map, if you want to find, let's say, certain virulence factors or certain other genes, you can do that. Now, can you maybe reduce your font size or something? It looks like the checkboxes that you have on the side are not aligned for some reason. So it's supposed to have up, down, and don't care. There are 3 checkboxes there, and somehow only 2 are visible there. I don't know why. Do control minus maybe. No?
No, yeah, I'm not really sure what's going on there.
Well, I'm seeing 3 checkboxes when I'm looking at it. Everybody has 3 except you,
Hey,
and you
that's
are the one
okay.
presenting.
That's okay. You guys, so long as you have the
Reload the
checkboxes,
page.
we're all good.
We need another checkbox. Reload the page. Yes. So the first thing I usually do, if you scroll down on the left-hand side, so there are filters available for log ratio and z-score, right? So typically, when people do the differential expression analysis, you kind of define your gray zone, the insignificance zone, right? So anything where your differential expression is, let's say, log ratio is between -1 and +1, you want to kind of like, throw it out, or it's not like, not much interesting, right? So those are some of the filters you'll apply first before you go to the heat map or you start looking into the gene in more detail.
So working off what Malik said, these are, by the way, absolute values. So selecting 2 right here would just filter this gene set down to only those that are upregulated or downregulated with a log 2 fold change of 2. So I'm just going to click filter. So that narrowed it down from 2,300 to 735 results. I don't think that modified the heat map though. I can reload the page and try doing the filtering first. Yeah, this looks like a smaller set. Okay, so just take a note, if you want to filter it down and then see those results in the heat map, you'll have to do that manipulation first before you do clustering or anything like that.
And regarding your question earlier, if you are interested in looking at the genes only, let's say transporter or virulence factors or something like that, you can enter the name of those genes or keywords or a list of identifiers in the text box over there where it says locus tags or keyword. So you can put, let's say, for example, transporter, or
I'm going to see
what
if
are
I
the
can
other
apply that
keywords?
and only have those show up in the heat map real quick.
Define your keywords. Throw some keywords here. Transporter is mine.
Any other fun ones? Synthetase, sure. Let's do some filtering. So there are 109 results that have those. Let's add log 2 fold change. So there are only 19 that have either of those 2 keywords. And those are going to be these genes. If you want to limit the heat map to things that only you're interested in, apply all of these first and then go over to the heat map so you can see those filter results.
So the other interesting ways in which can, you can have an interplay between some of the comparative genomics tools or other data types that we saw and your gene expression data heat map. So for example, you can get a list of genes belonging to certain pathway. Just copy the locus tags or copy the BV-BRC gene identifiers. You can post that entire list over here, and you can look at expression of only those genes. You can also take the genes that you have identified from doing, let's say, protein family analysis, where genes that are not very well conserved across multiple species or something like that, come with that list over here, and then you can look at the expression of those genes. So that's a good way to utilize learn about some data or genes, comparative genomics or other data types that we have, and understand what's going on with those genes in terms of gene expression.
So we have a question.
I'm using the same parameter that you're using, but I see difference in my map, slightly the clustering. So is there am I not doing the right?
Can you scroll down on the log ratio side? I think it's about the same.
It's almost same, right? But I see the difference.
It's probably the contrast. I think some of those green values that are there are just above 1. So it's looking like dark green. Here, it looks very close to black. I think it's just the gamma radiation coming out of the out of your monitor or something. It looks like.
In summary, RNA-seq is hard and we're trying to make it easier. Any questions? That pretty much summarizes RNA-seq unless I'm missing anything.
You mentioned the host HiSat pipeline?
Yeah.
Okay.
So I think there was one question over here which asked, can we see only statistically significant differentially expressed genes in the heat map? So I think one of the files that we have in the output from the DE-seq, it also gives you the p-value, and I think it will be useful when you have multiple replicates, especially because if it's just one sample, then it's not that interesting or powerful. You can use that file and maybe filter out the genes if you want, and then kind of like, cut and paste it here and then see only the expression of those genes.
That's also one of the future
So one of
improvements we
the-
have
so
in
one of
mind
the-
is to add a p-value filter so that you can
Exactly.
actually perform that type of.
Yeah, we were talking about the advanced clustering options. So you can cluster the genes and the conditions by multiple clustering algorithms. So there are about 8 different methods available, including Pearson and Euclidean distance and city block and blah, blah, blah. And then there are clustering type also, whether you want average linkage, complete linkage, and things like that. And I think the best way I can describe this is that it's like kaleidoscope. Every time you use a different method, you get slightly different pattern. And suddenly you find something that looks really interesting that you might want to take a screenshot and put it in a publication. But yeah, I mean, all of these methods will give you slightly different kind of like layout and clustering of the data. I personally like either Pearson correlation or the city block. Those are the ones that I kind of like, work better for me when I try certain data types. There is another question here. I think somebody made the beginning of the session and is asking, can you show the input page one more time? The person wants to use HiSat2 with 2 conditions and 2 replicates. So how would somebody do that?
Sure, I can go ahead and just do an example of filling that out. Sorry, it took so long to get to that question. So, Homo sapien. If you notice, selecting a eukaryotic genome will force you to select the host HiSat2 strategy and disables the rest of them. So that's why I recommended selecting your target genome first, because if you select one and then select this and then change this, then sometimes it will force select a different one. So we select that. Let's just select another folder. I'm not going to submit this job. I'm just going to fill it out, test, host, workshop. I mean, that's pretty much the only difference. When filling out the form, you would just select a eukaryotic host, and then it will auto-select the strategy for you. As before, you would just go ahead, fill this out, 2 conditions. I'm just going to select some random reads files here. Go ahead and define the contrast. And then you just go ahead and click submit. So doing a host run is pretty much identical with the exception of selecting a host target genome. Hope that answered the Zoom question. Marcus, do we have any more Zoom questions or is that all? What you can do is if you have a mixed RNA-seq sample is use the FASTQ utils to align those reads to a host or bacterial organism and save the unmapped reads. And then once you have the unmapped and the mapped reads, you can just take those reads and run the pipeline twice with a different reference genome.
I mean, you don't even need to do that, right? The moment you pick the reference, it's going to align whatever it's going to align, right? So,
Yeah, well, that's just to remove the excess unneeded information. It'll make your run faster, I think, if you were to separate the reads out first.
Skew and just show quickly one more time how to report a job and how to rerun a job.
Yeah.
Do you have any failed jobs there or do you want to?
Those are my jobs. So yes, they're a failed
Sort by.
job.
You can see all the failures here.
One last.
Select one of them, sure. And then after selecting a job, a failed job in particular, this report issue button becomes available for you. You can click on it.
Gives us all the information about your job, what parameters you use, where your input files are, when you ran the job, and all of that information, what exact parameters you use and things like that. Whenever somebody complains about a failed job, for developers, when a request comes to us, earlier we were having really hard time trying to understand where the data is, and we were asking 100 questions to the user. If you report it this way, we have all the information we need. Some of us, like the primary developers, we have ability to check your account as a super user. So we can impersonate any user to help you debug one of these failed jobs. So we do not go into your account to look at your data, look at your result or anything like that. The only reason we do that is if there is a question from you and if you need help. But when you do that, we can look at the file, we can see why exactly the job failed. We can look at the error output. And then based on that, we can either rerun the job.
Sure. So I'm just going to restart from the top. So at any point, you can just select jobs here in the bottom. So there's this little thing at the bottom that says jobs and uploads. You'll be able to see it if you look at the website and you click on jobs, it will take you to the jobs page. You can also click workspaces and there is this my jobs selection right here. And that will do the same thing and take you over to the jobs page. This is going to be a list of all the jobs you've ever run. You can sort it by the ones you're interested in. In order to report an issue, actually, you don't even have to click on a failed one. You can report your completed jobs too, but I don't think we get a lot of those.
Oh,
Nice
yes,
to
we do.
get some
Oh, yes, we
Successful
do.
reports. I've never seen a successful report, so.
We get them often because sometimes the job, the pipeline says it completed, like it ran through the pipeline. And it could be an assembly job and it didn't find any contigs because it couldn't create contigs because perhaps you submitted contig files, but it tried to run and it completed and it said, I'm done. I did what you asked. Not my fault, but it looks like completed to you and you click on it and you say, hey, where's my stuff? Then we have to describe it to you. One other thing, Clark, is could you click on a job, like let's say you did a phylogenetic tree job and just click on that line and then click on rerun. Okay. So let's say you realized after you submitted it, it's like, oh man, I wanted to add this other thing to it. So you can click on that and it'll save the job where you were. Go ahead and click on rerun. And then you could add something to it. It doesn't have the title, it doesn't have the folder, but it saved all that stuff. And this is especially useful if you've gone in and added hundreds of things and you forgot you needed one more, but you'd already started it. Or maybe it had finished and you don't want to load all of those again. So that way it will do it for you.
So if you have done some analysis and if you want to redo it at some point, then you can just run the whole job again without going through the whole process of selecting your files and parameters and all. The other place that can be useful is, remember the variation surveys example that Rebecca showed, where you are using the same input data, the same conditions and everything, but you want to just do the assembly or variation analysis using different algorithms. So in that case also, you don't need to keep going through all the steps again. You can just press the rerun button, come here, use a different algorithm, just change that part and then resubmit the job. Every time you come here, it is going to ask you to provide a new output file name, right? So that is one thing that you'll have to provide because you don't want to keep overwriting your previous output.
I don't
That's
think
the
we even
only
let
thing
them
that is not
overwrite
out of
previous output. You will not be able to do that.